So this is the overview of bioinformatics lecture. I'm Nima, bioinformatics is my research area. Okay. So what is bioinformatics? Any guesses? And Kevin, you can type in the chat. I'm monitoring it. Any thoughts? What is bioinformatics? Yeah, go for it.
Using like information you get from like biological data sets to make like conclusions.
Nice. So I like so there's like some notion of information, so informatics, information, some notion of biological data. I like it. Any other guesses? No, that's fine. Cool. So I don't have any concrete definitions either, so I just Googled it. So one definition from Wikipedia, bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data. Another definition that I found is the collection, classification, storage, and analysis of biochemical and biological information using computers, especially as applied to molecular genetics and genomics. This is from Webster. And this third one, this is one that I kind of personally like the most out of these three. It resounds with me. Bioinformatics is conceptualizing biology in terms of macromolecules and then applying informatics techniques to understand and organize the information associated with these molecules on a large scale. And the reason why I like this definition is that I think with bioinformatics, sometimes we lose sight of, we're really describing these biological phenomena. The real world is complicated. You cannot capture all of the complexity of the real world on a computer. So in bioinformatics, we're kind of like taking the real world biology and trying to like model it, conceptualize it in computational terms. And then we use standard computational techniques once we've transformed into that space. And then hopefully we can then use that information to make some interpretations about the real world. So here's kind of my definition of bioinformatics. Imagine that we have like this line over here where on one end we have biology, on one end we have computer science. And then if you imagine I kind of like draw a line right down the middle, in my opinion, I feel like bioinformatics is whatever's on the right side of this number line. And I feel like computational biology is whatever is on the left side of the line. So I feel like bioinformatics is kind of like you're more of a computer scientist that develops tools for studying biological data sets. And in my opinion, computational biology is really like you're a biologist who uses computational tools like you would any other tool in your lab. So you use, pipettes and gels and whatever, and you also use computers. This is not a good definition. Where does chemistry fit into this? Where does physics fit into this? Where does statistics fit into this? So there's not really one definition. If you ask a million people, you're gonna get a million different answers. But yeah, just some mixture of biology and computer science and stuff like that. Any questions so far? All good? Okay, so first we're gonna do some intro bio just to bring everyone up to speed. And then we're gonna transform those into some computer science problems. So first, the central dogma. That sounds fancy. So what's the central dogma? Has anyone heard this term before? Yeah, go for it. Anyone heard this term, central dogma? Any thoughts? Yeah, go for it. Yeah.
DNA replication.
Ooh, so DNA, it's not replication, but you're close. So it has to do with DNA, absolutely. Some process that DNA goes through. Any thoughts? Transcription, that's one of the steps, absolutely. So what is transcription? You don't know, that's all good. So DNA is one of the words that's involved. Transcription is one of the words that's involved, absolutely. I think there's 3 more words that are involved.
Is that DNA is like the base of all living things?
DNA is the kind of, yeah, in a way. So DNA has like all the information that kind of is behind. It's like the instruction manual for all living things, absolutely. So, yeah, so DNA, transcription, those were 2 words that were said that I agree with. RNA, that's a 3rd one, absolutely. There's 2 more words. If anyone had to guess. No? Okay. Give us a hint. Think about gains. Protein is one of them. Yep, so I'll go from there, perfect. So all the gains, what? Oh, no, you're good. Yeah, so the central dogma of biology is kind of the forward in motion propagation of information in the cell. So we have DNA, which kind of codes for all of the instructions for how to do anything in our bodies. Then there's a process called transcription, where we create a copy of a gene from DNA to RNA. So we're kind of transcribing DNA to RNA. And then we have a third step called, or a second step to create a third molecule. We have a step called translation, where we're taking an RNA molecule, specifically mRNA, messenger RNA, and then we're translating it into protein. And protein is kind of the functional unit of the cell. Proteins are like the smallest things that do stuff in general. Everything in biology, you can assume there's always going to be a counterexample. So everything that I say, take with a big grain of salt. Because it's actually not that simple. Sometimes proteins can replicate themselves. Sometimes RNA functions as RNA and doesn't get translated. We can have reverse transcription. We can have replication, blah, blah, blah. But for SPIS, we're just going to keep it simple. That was a little scary. Keep it simple. DNA, RNA, protein. DNA to RNA is transcription. RNA to protein is translation. Okay. So what is this? And kind of the analogy that I like to use is you can kind of imagine imagine that there's like a library where you're not allowed to take books out, but you're allowed to like read any books in the library, and you can make copies of any pages of the book. So imagine that there's a book in this library that has the instructions for building whatever you want to build. So DNA is kind of like that instruction book. It has every single set of instructions for every single thing you'll ever want to build, but you can't take it with you. It has to stay inside the library. If you photocopy the instructions for making like a bike, for example, one specific thing you want to make, you can copy the instructions for it, and you can take that copy with you out of the library. That would be RNA, basically. And that copying process is transcription. It's not the exact copy. It's kind of like a photocopy that you made that's a little bit, maybe it has imperfections or whatever, but it's a copy that you could take away with you that you can then build your bike out of. And then protein would be like the bike. So basically actually building the bike from those instructions I copied, that would be translation. And then just, I guess, one more trivia. In this analogy that I have, what is the library? Is that like, is there a part of the cell that folks know the name of? That the nucleus, exactly. So DNA cannot leave the nucleus, but RNA can leave the nucleus and then do all the fancy stuff. And that's kind of the extent of my biology knowledge. So we'll pause there. So let's zoom in on each of these. So transcription is the first process that I talked about. So transcription, DNA is transcribed to RNA. DNA is a double-stranded molecule. A pairs with T, C pairs with G, and then RNA is a single-stranded molecule. So for the purposes of SPIS, we can kind of just assume DNA is some string over the alphabet A, C, G, T. So you all learned about strings. Welcome back. You all learned about strings. For the purposes of SPIS, DNA is just some string over the alphabet A, C, G, T. So you can only have the letters A, C, G, or T, but otherwise it's just a normal string. And really the A, C, G, and T, they just represent these molecules, but we don't have to worry about them. RNA, you could think of as a string over the alphabet A, C, G, U. So there's no T, T gets replaced with U in RNA. And kind of the biological mechanism that this happens with is, you can kind of imagine this green line is a piece of DNA. This little arrow thingy is called the transcription start site. The area before it is called the promoter, and this like red line is the transcription end site. We have these proteins called transcription factors that they like kind of, they're just floating around in the cell, and they just kind of like just stick there. They just stick to, oh, actually, I guess Kevin can't see. So yeah, they're just kind of floating around, and they just kind of stick to that promoter region, the section just before the transcription start site. And then there's another protein that is called RNA polymerase that it's also just kind of like floating around in the cell. And then it just, when it sees a transcription factor bound to the promoter, it just binds near the transcription start site. And then it kicks off transcription. It basically goes letter by letter by letter in the DNA molecule and then says, oh, the current letter in the DNA molecule is A. I'm going to create a T. Then the next letter is a C. I'm going to create a G. And it kind of builds this molecule of RNA piece by piece by piece by piece by piece by piece until it hits that transcription end site. and then it's done. The RNA molecule just kind of detaches and floats away. Other stuff happens, but you don't have to worry about that. Yeah, so as a brief summary of what we as computer scientists can pretty safely assume, and this is actually a very safe assumption until you get to like really, really niche bioinformatics stuff. So pretty much anything that you'll ever do in bioinformatics, you can keep this assumption. DNA is a string over the alphabet ACGT, and you can think of transcription as just being the process of copy and paste that string, but just replace all the T's with U's. Like an overarching summary that we'll have. And in reality, it's actually a little bit more complicated. There's what's called splicing, where pieces get cut out. There's a 5' cap. There's a 3' poly A tail, whatever that means. Don't worry about it. For SPIS, DNA is a string. We copy and paste it and just replace all the T's with U's. And that is transcription. Questions kind of make sense? Okay. All right, what about translation? So translation, I told you RNA is translated into protein, and really it's messenger RNA that's translated into protein. So RNA is a string over the alphabet ACGU. Protein, we can also kind of think of as a string over the amino acid alphabet. So there's 20 amino acids. You might be thinking, well, how do I go from 4-letter alphabet to 20-letter alphabet? There's this translation codon table. So RNA, we actually, every 3 letters of RNA, every like 3-letter chunk, translates to a specific amino acid. That's why they call it translation. We're kind of going between alphabets now. So the way to read this table, this kind of the big rows represent the first letter of the codon. Codon is just a word for a 3-letter RNA chunk. The columns represent the second letter, and then the smaller rows represent the third letter. So for example, AUG codes for the amino acid methionine. Okay. So some things that I want to highlight here, there's this AUG that's highlighted in green. There's also those 3 things that are highlighted in red that say stop. Translation starts with an early on AUG. So if I have this RNA molecule, translation starts with one of the early on AUGs. And then starting from that AUG, I translate each 3-letter window piece by piece, looking it up in that table, and then converting it to the corresponding amino acid. And then the moment I hit one of those 3 red stop codons, then I stop translation. My protein is done. Okay, so how does this look like? I have an RNA sequence. I told you translation starts somewhere at a pretty early on AUG, which itself codes for methionine M. And I just go letter, like codon by codon. I slide over 3 letters, translate that. Slide over 3 letters, translate it. Slide over 3 letters, translate it. And each of these I'm just looking up in that codon table. Keep going, keep going, keep going, keep going. Oh, UGA is a stop codon. So then I would stop there. And this is my final protein. So here I just have a figure showing the structure of RNA. The coding sequence that we looked at, this Matthias part, this is this green window. The stuff before it, this is called the 5' untranslated region. The stuff after it, this is called the 3' untranslated region. Don't worry about what those mean. Yeah, go for it, yeah. It's one big string of amino acids that then folds on itself, which is a little bit weird. So you might have heard of this thing called like protein folding. There's like a tool called like folding at home or something like that. So translation creates this linear amino acid string. It's just a linear molecule that has amino acid, amino acid, amino acid kind of in a single chain. That's called the primary protein structure. Then those amino acids fold onto each other into these 2 shapes. They're called alpha helices and beta sheets. And then those alpha helices and beta, and that's called the secondary structure. And then those pieces fold on each other to create these crazy protein structures. That's the tertiary structure. And then individual folded proteins might come together. So like in this case, this is a protein that actually 3 copies of the folded protein fit together to build a complex. And then that's like how the protein actually functions. And this problem of like, given a sequence, what is it going to look like in 3 dimensions? This is like a very hard problem, but we're not going to worry about it too much because this is scary. So we're going to keep it simple. So this is the extent that we're going to care about, but just I want to expose you to all the complexities that in the future, maybe that'll be something that's interesting to you. Any questions? Kind of make sense. And yeah, please always feel free to interject with questions. I can like derail the conversation wherever we want to go. Yeah, go for it. Yeah. Good question. So why is M, so AUG translates M, why is this the start codon in like most eukaryotes? So like humans are eukaryotes, yeast is eukaryote, like most animals are eukaryotes. We don't know. And then it turns out actually in some bacteria, there's other start codons other than AUG that also we're like, I don't know why this happens, which is why like in biology, the one fundamental truth that you can always trust is that anything that you think is a rule, there's going to be counterexamples. And then why are those 3 the stop codons? I don't know. I think it has to do with how like the mechanism of the RNA, no, not polymers, of the, I forgot what protein does the translation. I think it's something to do with how that machinery of the cell hits the codon. So if you look at the codon table, there's like, you'll notice that codons that code for the same letter usually have the same first 2 letters, and then the third one might vary. This is because the thing that binds to the codon to recognize it strongly binds to the first 2, and then the third one's sometimes called like wobble. It doesn't really care as much. So I'm assuming it's something similar to that for the stop codons, but I don't know. Good question for a molecular biologist. That's the beauty of being in bioinformatics. When computer science people ask me stuff, I can say, oh, I'm a biologist, I don't know. And then when bio people ask me stuff, I'm like, computer scientist, I don't know. So we could take advantage of both worlds. But yeah, good question. Short answer is, I think most scientists just don't know. It just kind of is. Yeah, bio is weird. Cool. Okay, so another thing that I want to quickly introduce is what's called natural selection. So this is probably something folks have heard before, but like if you had to summarize, like what is natural selection? Survival of the fittest. I like that. Okay, was that were you going to say that verbatim?
It's like I heard someone else say it.
Oh, okay, you're nodding along. Perfect.
We were talking about this morning.
Uh-huh.
About how like Asian genes are like shorter for height.
Not necessarily. So actually, yeah. So that's actually a misconception. So it's actually largely because of diet, and then actually when like a lot of more nutrient-rich things were introduced to Asia. So like, I don't know if you've heard of like yellow rice. It's like a genetically modified rice that has a bunch of nutrients. Then like a bunch of growth spurts happen. So actually not necessarily, but natural selection does, like, why does a giraffe have a long neck? Like that is something that like plucking the leaves at the top of the tree was more beneficial. Yeah. Yeah, go for it. Yeah.
Survival of most adaptable, maybe.
Ooh, survival of most adaptable. I like that. Any other thoughts? So survival of the fittest what? The fittest DNA, okay. Yeah, go for it. Yeah. The most favorable traits, I like it. I like it. Is it any trait? Or is it a specific type of trait? Now I'm getting like kind of niche, so it's okay if no one knows it. Yeah.
Traits that are best fit to their environment.
I like it. So traits that are best fit, but any possible traits, or are there like specific traits that count for this?
Dominant.
Not necessarily dominant. Recessive can be. So I like that. That now we're getting to like Mendelian genetics. Yeah, go for it. Did you have any?
Genotype.
Genotype. So it's actually we're related to phenotype because phenotype's what functions, but close. So why did you say genotype?
It's like what being passed down.
Passed down. So heritable traits. Absolutely. So that's the niche that. So like if for some reason one day like I don't know your arm gets cut off and then some totalitarian regime is like I'm going to kill everyone that has 2 arms. I don't know. This is a weird example. That's not a heritable trait. Like you losing your arm was not heritable or whatever. so it's only heritable traits. So that's why I wanted to kind of ask about the genotype point that you brought. So now to kind of pick up on, so what is the word genotype? What is the word phenotype? So there's always natural variation in a population. All of us look different. Any species that you look at, there's natural variation in the population. And this variation is both genotypic, so both on the level of DNA, and phenotypic on the level of just physical features. Some physical traits are derived from your genes, some are not. So, you look for the most part like your parents, but you don't look identical to them. There's other things that are not, there's some things that are not heritable traits that you also get. So yeah, natural selection, it's this notion that like traits that improve the fitness of an organism will cause that organism to be more likely to reproduce. So any traits that are heritable will pass down to the offspring, and then individuals without that trait are less likely to reproduce. So then in the next generation, a larger proportion of the population is going to have that beneficial trait. And I have an example that this is tied from actual history. I'm not a very good artist, so we're going to kind of go back in time to the Industrial Revolution in like the 1800s. Shit, I think I failed that AP test. I think it was 1800 something, I don't know. So Industrial Revolution, though, like in England, they basically had these factories that could now build things at super big scale. But these factories would like just blow soot into the air, and that soot is going to hit the trees, and there's animals living on the tree. So here in this diagram, this beautiful rectangle, this is a tree. So suspend your disbelief. And these beautiful circles, these are bugs. Right? Okay. So each of the bugs, they have natural variation in just what color they are on the surface. And the tree has some color. So why do you think maybe these lighter ones, there's most of them in the population?
They blend into the tree.
They blend in, right? So if a predator is trying to eat them, it's hard to see these light ones, but it's easy to see the dark ones. But let's say industrialization, capitalism is evil, all that stuff, they're burning the world down. Soot goes onto the tree and it makes the tree darker. Well, now these lighter ones, they're not as blended in. Now we're kind of like in the middle of the light ones and the gray ones. So maybe both of them are roughly equally fit. So now the light ones aren't as beneficial. They do get picked off a little bit. Now, even more evil capitalism, the tree gets even darker. Now the light ones actually stand out, but the ones that used to be the middle color, which was less fit before, now these ones are more blended in. So maybe in the next generation, a bunch more of the light ones get picked off. And the next generation, we have just more of these middle colored ones. And then, global warming, the planet's dying, it gets even darker. Now maybe the darkest ones are the most fit because they blend in the most. So we actually had a complete shift from favoring the light ones to now favoring the dark ones. And this is kind of assuming that the color of the bug is genetic, or it's somehow connected to the gene. That's a heritable trait. Okay. So the key takeaway is if a trait is essential to an organism's survival, it's going to be conserved in the population. There's a reason why, all of us have slightly different hairstyles or hair color or whatever, but we all have 2 eyes and 10 fingers. And, there's kind of some standard things that, all of us have a heart, all of us have 2 lungs, 2 kidneys. They're like some very standard things that you need for survival. If something happens that kind of kills that trait, anything that has the kind of mutated version that doesn't have that trait is going to die off. So as a result, anything that you observe, I know it's like very dark, but anything that you observe, there's kind of this survival bias implied where you can't observe things that don't get to survive to be observed. So conservation, if you notice that something pops up a lot in this given species, that might be important for its functionality. I work in viral epidemiology. So for me, a lot of times this is looking at like in the spike protein. If, with COVID-19, it's been spreading. If we see in the spike protein or like wherever in the genome sequence, mutations are occurring across the, viral population, if there's certain regions that aren't mutating, that tells us, oh, there's probably something important going on here because anything that did mutate there didn't survive for me to be able to observe it, if that kind of makes sense. So just kind of in the back of your mind, kind of, this is not the definition of natural selection, but as a product of natural selection, this kind of persists as well. Any questions? Kind of makes sense. Okay. Okay, so now we have all the bio background that we need. So you're all biology experts. Perfect. Easy peasy. Okay, so now we're going to start thinking about some bioinformatics problems. So the first problem that we're going to talk about is what's called sequence alignment. And the general idea here is if I have two strings S and T, if I were to able to like kind of stick gap characters, stick like spaces in any position in either string, can I make those two sequences line up better? And here's just a kind of concrete example for folks to look at. So here's two strings. At a glance, they don't seem to really line up that well. So if I gave you the ability to stick spaces in either string, could you make these line up better? So maybe take a little bit to think about it. And if anyone wants to kind of give me some suggestions. So yeah, if I stick spaces in them, so think of this like a puzzle. How can I make them line up? Yeah, go for it, yeah.
Space between like the top line, the first A and first G.
So you're saying put a space here, so that's going to shift over the G, shift over the T, shift over the A, shift over the C, shift over the G, shift over the T, shift over the A, shift over the C, shift over the G. Okay. So yeah, actually, yeah. So there's kind of two spaces that I could put to get things to line up really well. One of them is precisely right there. By sticking a space here, I pushed all of these letters over one. So now, even though they didn't used to line up, just adding this one space makes them line up really well. If I put a space here, now even this T lines up at the end. So why do we care about this? Well, in practice, you might know that like humans have a lot of things conserved with like, I don't know, mice and with yeast and with all sorts of other organisms. So like, let's imagine that this is like the gene sequence of like hemoglobin. So hemoglobin is a protein that's important for carrying oxygen in the blood. So like if I'm trying to compare the evolutionary history of hemoglobin between mice and humans to see like what parts of that genome line up, mutations can happen, which could either be substitution. So you'll notice over here, this is not a perfect match. There's some mismatches between these two. But then there's also some spaces. So kind of the biological motivation here is in actual mutations, there's kind of three types of mutations. There's actually more than that, but for the sake of simplicity, let's think of like three types of small mutations. There's substitutions, where you have one letter turn into a different letter. There's insertions, where you stick in a new letter. And there's deletions, where you delete a letter. So across the evolutionary landscape from some common ancestor to human and some common ancestor to mice, these mutations happen in this gene, and we want to be able to figure out, based on just the sequences, where did those mutations occur? So if I can line these up, then I could say, oh, this is probably a substitution, and then these two are either insertions or deletions. That kind of makes sense. And then by doing that, we could see like, oh, what parts of these two gene sequences line up and are conserved? And that's probably important for functionality. Okay, so let's translate this into a computational problem, because this is SPIS after all. You see me ramble on about APS. We like computational problems here. So if you, so the first thing that we want to do is just, if I gave you one of these alignments, so if I gave you these two sequences with the spaces in between them, how would you score them in the first place? So basically what I could do is, if you gave me these two, if you also gave me a gap penalty, so I don't want to put too many spaces because spaces are kind of cheating. So I want to have a little bit of it, but I want to have a penalty if I use one of those spaces. And if you gave me a scoring matrix where you basically tell me what is the score or penalty of having one letter line up with a certain other letter, I could define the score of an alignment as just being the sum of the scores of the columns, where each column is either the gap penalty, if I have a gap, or it's a value that I look up in that table. So that's kind of an abstract mathematical definition. It makes a lot more sense if I just look at an example. So here's an example alignment. Here is that matrix that I mentioned, where maybe the rows show me what's in one column, in one of the rows, and then the columns represent what's like in the other sequence. So basically, like, this is if you had a C in one of the strings and an A in the other one at a given position, versus like A paired with A versus like T paired with C, whatever. And then here's my gap penalty. Okay, so based on this, what do you think? What do you think is the score of, let's see, where am I? Of the first column? So I have an A and an A. I see a 1. Exactly. So I have an A in one column, in one sequence, paired with an A in another sequence. So that's a +1. What about the next column? -1, how come? Because there's a gap, exactly. So here I have a gap character, so -1, I get a penalty. And the next column,
One.
+1, so G paired with G is +1. C paired with T is 1, 1, 1, 1, 1, 1, -1. I have a C paired with an A here. -1, and then +1. And the reason why I'm showing you this as a matrix is because if I knew something about the biology, I could make these values different if I wanted to. So here I just kind of have a match of +1 and a mismatch of -1, but I could have made it more complicated if I wanted to. So the score of this alignment is 6. So with sequence alignment, what I want to do is I don't know where these spaces are. All I know is if you gave me an alignment, I could come up with a score. And the question is, can I come up with an alignment that returns a maximum? Can I come up with an alignment that maximizes this scoring function? So when they started, there was all those mismatches and stuff. So this probably gets a really bad score. And I want to be able to figure out where do I put those spaces to maximize this scoring function without any other information. So there's a couple different versions of this problem. One of them is called global alignment, where basically given 2 strings, I want to return a maximum scoring alignment between them. So I want to basically have the entirety of the first sequence and the entirety of the second sequence, maybe with some spaces stuck in between them. There's a similar problem called the local alignment problem, where basically given 2 strings, I want to find a maximum scoring alignment of any substring of the first string with any substring of the second string. So this is called local alignment. So here, maybe having the entirety of the 2 strings doesn't end up in any good alignments, but if I just cut out this substring and this substring out of all possible substrings I could have cut out from either sequence, this gets me the best alignment. And then there's also called the multiple sequence alignment problem, where if you gave me a bunch of strings, how do I do it across all of them? So this is something that we do with like COVID-19 sequencing, where if you gave me all of these genome sequences from around the world, how do I line them up accurately? Okay, and I'm not going to talk about how we do this. I just want to introduce a bunch of bioinformatics problems. And if you're curious, I have a bunch of resources for all these. So like for this, for COVID, I actually came up with the fastest tool to do this. I can send you all the paper, just little plug, humble brag. Yeah, go for it. Yeah.
So, like, when you find all these like weird combinations or whatever, right? Like, what does that mean to you?
Oh, to me or to
So, like,
an epidemiologist?
whoever.
Because to me, I have no idea. I basically crunch the numbers. I'm like, here you go. Job done. So it depends. So like, let's pretend that these are, I think here, these are all hemoglobin of different species. But like, let's imagine, actually, let me see if I can find it. Do we have a good, whoops. Let's see. And I know that the screen's not sharing, Kevin. I'm going to share it in a second. Whoops, what are my, okay, let's see. So, I don't hard to see. I'm going to maybe pull this over here. Oh no, my slide stopped sharing. Darn it. Presented view. Oh, I should have just made this like show as tab. That would have been smarter. I am dumb. Okay. There we go. Okay, so let me pull it up in this tab so I can actually see the screen. Do we have like a global alignment of it? Let's see. Oh, maybe we don't. I'll show you one of mine. Where did I do this? Oh, actually, maybe I'll show you another resource. So there's a tool called NextString that I think visually highlights Oh, and now I'm going to share my screen so that Kevin can see again. There we go. So there's a tool called NextStrain that you can basically, they have a few different viruses of interest, but if we go to COVID-19, loading, there we go. So this is like an evolutionary tree of the COVID-19 sequences that we have, which I'll talk about a little bit later. So this is the genome sequence of COVID-19, and then don't worry about what this plot here means, but basically what we could theoretically do is if I lined up a bunch of COVID-19 genome sequences, I could see what parts of the genome are mutating versus what parts aren't mutating. So like this plot, actually maybe I will describe it. So this plot on the vertical axis, this is saying there's different letters at this position. So a big value means there's a lot of different mutations there. A small value means there's no mutations. It's conserved, which what does conserved mean?
It's essential.
It's essential to functionality. And if you just look at this, you might notice, so spike protein is mutating a lot. This is actually essential for its survival to mutate because, so spike protein is how the virus invades the cell. So your body's kind of defense mechanisms against it fight it. So it has to mutate to evade your defenses. Versus other parts of it, like this ORF1AB, you'll notice there's like barely any mutation. So it seems like this needs to exist as is for its survivability. And then the rest of it kind of has normal mutations. So like this one being hypermutated means like, oh, this is like trying to evade something. And then this one being like super low mutation kind of hints to us, oh, this is just generally essential for survival. And everything else is just kind of like randomness. Yeah, it's crazy. So I mean, this is helpful for like, if I want to design a vaccine, I should probably design a vaccine that targets how it invades the cell, but ideally a region that doesn't mutate very much. So you might have heard of, I don't know, like, has anyone heard the term like S dropout? Is that something? Oh, you heard it? So what is it?
I've heard of that. I don't know what.
Sure, no worries. So if you do a COVID PCR test, the way that they do it is they have what are called probes that, like they're called primers that are specifically targeting certain parts of the genome. And then if you basically, they try to like amplify any DNA or RNA that matches that sequence. And if it succeeds, that means something in your sample matched to it, AKA there is COVID in this sample. If it fails to replicate, that means nothing matched to it, AKA negative test. But it could be that if you target this region here that hypermutates, if this lineage has mutated in a way that that primer no longer recognizes, even though there's COVID, the primer fails to amplify and you get a negative test. So that's what the S dropout meant. If you Google it, it was basically lineages of COVID that like had mutations that then failed to replicate certain primers that we had. So yeah, so this stuff, it's useful for designing like COVID tests or in general, like any type of viral sequence test. It's useful for like designing vaccines, because if I also know information about how it invades the cell, maybe I can target an important region, but a part of it that doesn't mutate very much. I don't know, all kinds of stuff. Cool stuff, yeah. Okay, I'm gonna jump back to the slides. And I do a lot of like viral computational research, so feel free to hit me up if you have any questions about that. Okay, so I'm going to introduce the next bioinformatics problem. It's not related to viruses, but it's again related to humans. Most of these are going to be related to humans, but I also do a lot of work with viruses. But just most bioinformatics people do human stuff, so it's going to be a very human-centric lecture. Okay, so variant calling. This is the next thing I want to talk about. Has anyone heard of this term at all? It's all good. I'll just jump in. So have folks heard of like 23andMe and stuff like that? Yeah. Okay, awesome. So that's basically what they're trying to do. So no matter how diverse we all look, all of us have 99.9% identical genomes. And it's actually interesting that all of this diversity that we have is caused largely by that 0.1% that we differ from each other. So variant calling, so these variants, these are the parts of the genome that are variable between different people. They're the parts, they're that 0.1% that differs between people. And there's a few different types of variants that we can measure. And this is unrelated to COVID variants. That's an unfortunate, like they adopted that terminology without it being correct. So that's a whole rant for another day. Variant just means this. What they should have called it is lineage, not variant, but whatever. So yeah, so single nucleotide variants, these are individual letters that some people have one version versus some people have another version. So like here, this specific position, a lot of people might have a G versus a lot of people might have an A. In this position, a lot of people might have an A, a lot of people might have a T. So a single nucleotide variant, a single nucleotide that varies. We can also have structural variants, which are kind of things that either have insertions, deletions, copies, rearrangements, all sorts of stuff. So here, these are what are called repeats. This is actually a famous repeat, but I won't get too far into it. But like different people might have different numbers of copies of this repeated CAG pattern. Okay, so how do we actually determine variants? Well, what we could do is if you gave me like their DNA sample, I could sequence that DNA. We have a high confidence human reference genome that I can compare them to. So using that sequence alignment problem that I talked about a second ago, I can take all these short fragments of DNA that I've sequenced and align them to the reference genome, and then use those alignments to determine for each position of the genome, what is this person's genotype at that position. So like maybe this person, I noticed they match the reference genome this whole area, but at this position, roughly half of the reads have a T, roughly half the reads have a G. What might that mean? And this is now, again, testing biology information. Why might someone have like kind of half and half at a given position if I like just blindly sequenced all their DNA? This might be a little bit niche, so no worries. Yeah, go for it. Not necessarily. So dominant and recessive, like the reason that those exist is somewhat related to it. But what does dominant and what does recessive mean, if you remember?
They're like different forms of the gene.
Different forms of the same gene. And what's like special about like I don't even know how to phrase this question. You're close. So basically, hold on to that thought. So basically, dominant versus recessive, these kind of play a role because maybe I'll just jump to it just for the sake of time. Oh yeah, go for it. Go for it.
Your parents have different. That's specific.
Your parents had different versions. Why does that impact you? Exactly. So humans are what we call, and this is related to dominant versus recessive. Humans are what we're called diploid. We have two copies of each of our chromosomes, one from our mom, one from our dad. And then the whole dominance versus recessiveness is at some position. If you have a dominant version of a gene and a recessive version, the dominant one's the one that shows. But regardless, like the reason why that's possible is because we have two copies of every chromosome. So this position, if I observe this could mean that, oh, well, I have two copies of this gene, maybe one copy of it from one of my parents has a T at that position, maybe another copy of it from the other parent has a G in that position. And then this information, we can then answer questions about like ancestry. So you've seen 23andMe, so that's how they explore those types of questions. You see like, oh, in this population, most people have a G and you have a G, therefore you're part this population. It's also important for like detecting predisposition to disease. So I don't know if have folks heard of like BRCA, like BRCA. Is that so? You're nodding. So it's no, that's okay. I get excited when people nod. Sorry. BRCA. So BRCA, it's a specific mutation that, like at this position, if you have like a certain letter, I don't remember what letter it is, women are much more predisposed to breast cancer. So a lot of times they'll do like genetic testing, and if you have that specific mutation, then they'll recommend like certain preventative measures because like it's a significantly higher risk if you have that. So bottom line is this is really relevant to like not only ancestry stuff, but also like disease prevention, disease kind of studying and all that good stuff. There's some challenges. Some regions are difficult to sequence. The sequencing technologies themselves have errors. We have sampling errors, so we might not actually observe all the reads from a given person. So when we develop bioinformatics tools to do this variant calling, we have to kind of take into account the real world imperfections that we have. And again, I'm not going to talk about how we do it in bioinformatics, but if you want to learn more, I can send you some papers about like what are some tools that people have come up with. There's a bunch of different algorithms to do this. So let's imagine that I do this for every single person in this room, and maybe I do it like for the entire American population or something like that. What can I do with all of this information? There's also what are called genome-wide association studies. So maybe I want to see like, how did we even discover that this specific gene having this mutation is correlated with breast cancer? We could do these genome-wide association studies and see like, oh, a bunch of people that have this condition have this mutation, and most people that don't have this mutation don't have this condition. So we can kind of see what mutations are associated with what diseases. We can track genetic ancestry. We can do genetic counseling, all sorts of stuff. Okay. All right, I'm going to whiz through these like real quick because this is the second to last section. So another thing that we could talk about is called differential expression analysis. And I know I'm just throwing like a fire hydrant of information at you. I'm just trying to expose you to a bunch of stuff. And then if any of this sounds interesting, I can kind of send you more stuff specific to that in the future. All right, so differential expression analysis. Every single cell in your body has the exact same genome, roughly. Roughly is a caveat because, again, in biology, everything has counterexamples. But generally, every single cell in your body has the exact same genome. Yet your hair cells and your skin cells and your heart cells and all the different cells in your body, they look very, very different. So how is it possible that all of these cells that started with one cell that then became two, that then became four, and all have the same exact genome, how do they look so different? And importantly, how do they function so differently? And the reason for this is even though they all have the same genes and the same genome sequence, there's differential expression of genes. So remember I mentioned transcription. We take DNA and transcribe it to RNA. Then we have translation, RNA translated to protein. That is gene expression. A gene that gets translated is being expressed. So different cells have different genes that are being turned on or off at any given time. So the biological question here is if you gave me two different samples, maybe it's like skin cell and a heart cell, maybe it's a healthy cell and a cancer tumor cell, whatever two cells that have the same genome but look different, what are the genes that are differentially expressed between them? Ideally, as I mentioned before, proteins are kind of like the main workers of the cells. So ideally, I want to be able to measure protein level, like protein levels of each of the genes, but I can't do that in high throughput because of technical limitations you don't have to worry about. But because of the central dogma, RNA is a pretty good proxy. So I can measure RNA levels in high throughput. It's close enough to protein levels, so good enough. And the way that we do this is we reverse transcribe RNA back into DNA. We sequence the resulting DNA using one of these sequencing machines. Oh, and by the way, so this is called an Illumina HiSeq machine. Illumina is like the DNA sequencing company of the world. And I think from here, we might actually be able to see their office. It's like a big, actually, I think it's like a little bit further south. If we're on the other side of the building, they're like literally just down a couple streets, which is like insane. The biggest DNA sequencing company in the world is like in our backyard. So that's pretty cool. So yeah, UCSD, our bioinformatics program has a lot of collaborations with Illumina for obvious reasons. They want smart people, we want money and devices. Okay, and then after that, most of it is the exact same process. We basically map each of those fragments to the reference genome. And then what we can do is basically look at the reference genome and see, well, I know in advance where the different genes are in my genome, so I could just count how many reads came from this gene, how many reads came from this gene, how many reads came from this gene. I can count that for each gene for both of my samples. I can do some normalizations that you don't have to worry about. And then I could do some fancy statistics and see like, oh, this gene is much more expressed in one sample versus the other, or much less expressed or whatever. Okay. So I'm just going to kind of go through super fast. I think actually this is the second to last problem because I feel like I'm rambling for too long. So I'll go fast. Okay. So this problem, genome assembly, the question is, how do I even get the genome sequence? I kept mentioning we have a human reference genome sequence. How do we even get a human reference genome sequence? So like, have people heard of like the Human Genome Project from like, it's like from 2001 or something like that. So that was like the NIH project to try to sequence the human genome. So now we have a human genome sequence available to us, but how do we sequence one in the first place? Or like with COVID-19, this new virus came out, how do we sequence its genome? So because of limitations that you don't have to worry about, we can only sequence small fragments of an organism's genome. That's why all those previous examples I was saying, you have these small fragments that you're aligning to the reference. It's because I can only sequence small fragments of the genome. And what I want to do is be able to tie these short fragments together to then reconstruct the long contiguous genome that they all came from. So the computational problem here is if you gave me a list of reads, a list of these short strings, what is the shortest superstring of these reads? What is the shortest string that contains all of them as a substring? Right, because I could just concatenate them, but that's not very informative. So I want to find the shortest string that contains all of those little strings that I gave you. Give or take real world errors that we're not going to talk about. So there's a problem called genome assembly. Okay, and now the last thing that we're going to talk about is what's called phylogenetics. So phylogenetics, hopefully folks have seen evolutionary trees like this. Yeah. The rock, yeah. Gotta put them in. Harambe, RIP. So in an evolutionary tree, we have this tree structure where the leaves represent present-day species. The internal nodes, these internal things up here, represent ancestors. And the branches of these trees represent some unit of evolutionary time. So why do I say evolutionary time? Well, it could be number of generations. It could be millions of years. It could be days. It could be seconds. It could be number of mutations. Any metric where bigger metric means more evolution has happened. Okay. So we can come up with different models of tree evolution to describe like the probability distributions of the shapes of these trees. So like maybe we're asking questions like, are some tree topologies more likely to be observed? I don't know if folks have heard about like the whole like COVID origins, wet market stuff, maybe like raccoon dog. Yeah, so that was my team. So asking questions like maybe this scenario or this scenario, which one's more likely? We can come up with models of the tree shapes to ask which scenario is more likely to actually be observed. We can also ask questions based on models of sequence evolution. So this describes a probability distribution over observed sequences. So we might ask, are some sequences more likely to be observed than others? So this might tell us things about like fitness or whatever. And then a key problem that's of interest is phylogenetic inference. So if you just gave me sequences, how do I reconstruct an evolutionary tree like the one that I showed you on this page? Right, so this was constructed just from a bunch of sequences collected from all over the world, from like Chile, from the USA, from Vietnam. You know, if you gave me all of these sequences, how do I infer the evolutionary tree from those sequences? So the general structure is I take my raw sequences, I do something called multiple sequence alignment, and then I do something to get a tree from that multiple sequence alignment. The method that we typically use is what's called maximum likelihood tree inference. So if you gave me a multiple sequence alignment and you gave me a model of sequence evolution, can I find what tree out of all possible trees I could even theoretically look at, which one of them maximizes this likelihood function? Which one has the most likely chance of being the real tree to produce these sequences? Okay, so I've thrown a lot of information at you, so summary time, just because I know that everyone's probably super exhausted from just the fire hydrant of info. So what did we do? We started with some basic molecular biology review, and then we introduced a bunch of different biological problems and kind of discussed the computational problem formulation. I didn't give you the solutions. They exist. I'm happy to share that with you, but we just kind of started with the biology and then translated them into computational problems and kind of stopped there. The key takeaway that I think folks should hopefully have is bioinformatics means big data. So we're sequencing, like a single sequencing experiment from one of those machines generates like a terabyte of data, and we can run them every day. And if we have multiple of them, we can generate multiple terabytes per day. A lot of data. So we need efficient algorithms to solve those computational problems. So that's more of like the theory APS side of things. And we need optimized implementations of these algorithms so that, you know, the same algorithm, depending on how you program it, might run at different speeds. So that's more of like the foundations of computer science side, like the programming side of things. We have to have optimized implementations of these algorithms.